Or
matching the DNA of human being, right?
or any, yeah.
Here the DNA fingerprinting is related with the term fingerprinting.
Yeah.
Fingerprinting. As you know, our relation about the into the criminal investigations, what happens? Generally, the Department of Police used to fingerprint of DNA as a biometric record to find out the criminals, right?
Yeah.
But several times what happens, the fingerprint that can be matched with the other person. So due to the development of biotechnology, the scientists take the decision to find out the solid investigation criteria for a criminals.
Okay.
Generally what happens into the crime cases, the criminal do the crime and wisely what happens, he want to remove the all records, is give the means identification of the criminals. So regarding in that what happens, the one thing is common for investigating the criminals, that is their biological record. Also for finding out the biological record, the fingerprinting means avoided and DNA fingerprinting takes the place of investigation, right?
Yes.
Into the investigation, DNA fingerprinting, that is also known as the DNA profiling. It is also known as DNA profiling. For the DNA profiling, what happens? Who invented the DNA profiling? The name of scientist was the Alex Joffrey. He was the student of the University of Leicester into the 1985. And during his student time, he find out the criteria of the DNA fingerprinting. So it is the figure of the Alex Joffrey. The DNA fingerprinting that is based on the principle, okay, means first of all he take the prior criteria of the fingerprinting, that is known as the conventional fingerprinting. In the conventional fingerprinting of an individual, that comes from the fingertip and unique for an individual, right? This is used for the identification of person into the forensic lab and police station. But its major drawback, that is the conventional fingerprint, is that it can be changed by the surgery,
Yes.
right? So fingerprint means if you change this one surgery, okay, what happens? That all the record they are removed,
Yeah.
right?
Yes.
So in the most of cases, what happens after the surgery कि रिकॉर्ड हटाने के लिए क्या करते थे, वो इसको सब क्या करते थे, साफ कर देते थे, राइट? तो अब कैसा होता था, ड्यूरिंग दैट टाइम व्हाट हैपेंस, इट become means the case, complex case for finding out the criminals, right?
Yes.
So taking into the place, DNA fingerprinting was launched by the Alex Joffrey, and this is the very unique because this remains same in the all body parts, tissues and cells, as well as cannot be altered by any known method.
Okay.
If you see the second line, this remains same in all body parts. Do you see the your screen? आपको स्क्रीन दिख रहा है? तो इस स्क्रीन पे you see the second line, this remains same in all body parts, tissues and cells, as well as it cannot be altered by any known method.
Yeah.
This line remains that in all the cells of our body, DNA is present.
Yes.
So by the surgery, by the removal of the tissues, by the removal of the organ, it can't be replaced.
Yes.
Next thing, DNA fingerprint method, that is becoming primarily method for identifying an individual. Why? Because DNA is found into the every cell, and we can't remove the DNA from the cell. So a criminal carry the DNA which is same into the each cell of the body. And if we get the DNA from the crime place, then what happens? We can find out the person who do the crime.
Okay.
Right?
Yes.
Any doubt to this in understanding the principle?
Yeah.
Now see, what are the samples which is used for the DNA fingerprinting? The biological material which is used for DNA profiling, that is the blood. If you see the any crime place during the serials or during the paper, if you listen to it, read to it, then you'll find into the crimes, what happens, generally the blood, that is the main component of the sample, right?
Okay.
यानी
Yes.
किसी का मर्डर हुआ है, किसी का कहीं पे बात हुई, तो वहां पे ब्लड जो होता है वो मेन सैंपल होता है.
Yes.
अब क्रिमिनल कुछ भी करेगा, कहीं पर भी करेगा, तो कहीं न कहीं ब्लड जो है वहां पे प्रेजेंट होता है. But here is the most important thing. During this collection of sample as a blood, no RBC is used for the DNA profiling.
Okay.
What is used?
WBCs?
WBC. Why?
RBC might not contain DNA or might not contain adequate DNA.
No. Your answer is right. Not contain DNA, but your answer that is not correct, because into the RBC nucleus is absent. Hence DNA is absent.
Yeah.
Right? Your answer is right, but it is a partially right, because I am the professor of competition. So into the question, what happens? You never get the DNA is not present. Nucleus is not present.
Nucleus not present. Okay.
Okay? You are right.
Yes.
Now come on the second point. Hair, that is also important, because during the crime, sometimes what happens? The criminal, he removes his clothes, hankies, okay, during the crime, वो छोड़ देता है वहां पे, वो फेंक देता है वहां पे, right है. तो
Yeah.
so little quantity of the hairs, they are get sticks with their hankies, their shirts, their any other clothes, and that can be also become the sample for the DNA fingerprinting. Saliva
Yes.
is also important, because into the saliva, what happens? Sometimes mucus cells are present. Semen,
Yes.
semen is the also important sample which is used into the rape cases. Body tissues means the muscles like the nails, okay, teeth, they are also important for the DNA
Yes.
sample printing. And DNA samples have been obtained from the vaginal cells transferred to the outside of the condoms during the sexual intercourse. This one last case that has been, suppose among of all these, blood, hair, saliva, semen and body tissues, they are not present. And case is the case of the rape cases,
Okay.
right? So during that time, the condoms, they are used as a sample, because semen is present into the condoms, and from the semen, because in the semen the sperms are present and the sperm has the DNA. So these are the biological material used for the DNA profiling. Understand?
Okay.
Now
Yes.
next topic, in the principles, we have need to learn about the principle of DNA fingerprinting. First important point is the double-stranded nature of the DNA.
Okay.
DNA consists of the two strands. DNA is made up of two strands.
Yeah.
Both strands are anti-parallel with each other,
Yes.
right? The
Yes.
direction is opposite in nature. If we find out the one strand,
Yeah.
then we can find out easily to the second strand,
Yes.
as into the pen, right? These
Yes.
are the two parts. One is the head and second is the body. And
Yes.
here, if we find out the one part, we can easily find out the second part,
Yes.
right?
Yes.
Next important thing, most important, for variable number of tandem repeats. These are the fragment of the DNA whose position into different persons, right? It is nearby 20 to 80 base pair into the length. The most important tool for DNA fingerprinting. And Alex Jacobs' study of the myoglobins or muscle cells, he was found that location of VNTR, that is different into different individuals.
Yes.
So it never match, means you can see 99.99% V and TR location is different worldwide.
Okay.
So into this manner, the Alex Joffrey becomes success to find out the DNA fingerprinting.
Yeah.
After then, the starting procedure to create the DNA fingerprinting, right? The first step, the DNA is extracted from the nuclei of any cell in the body.
Yes.
It is the very most important step to DNA from the nucleus. As we go
Okay.
into the information, invisible inside the cell, nucleus is present. Inside
Yes.
the nucleus, DNA is present,
Yes.
and DNA is
Yeah.
found into highly coiled form. Problem to remove out the DNA. Right?
Yes.
The size of nucleus, that is the 10.9, 10.9.
Yeah.
Length of DNA, that is the total 2.25 meter in length.
Mm.
So that is the very highly coiled structure.
Yes.
How it become possible? Can you tell me कि कैसे ये संभव हो सकता है इसको?
Maybe with the help of enzyme, it's uncoiled.
Enzymes, right? So several here hydrolyzing enzymes are used, right? That enzyme cuts to the membrane, plasma membrane, nuclear membrane, and finally we separate out the DNA. That is the first step. After
Yeah.
separation, what happens? Washing procedure can be carried out, because
Hmm?
washing procedure, right? Washing is the important procedure, because DNA,
Yes.
when we obtain from the nucleus, that has some impurities like the nucleoplasm, ribosome, cytoplasm, and it can't be used directly into the DNA fingerprint,
Yeah.
right? For the
Yes.
washing, we use the hydrolyzing enzyme, right? Washable enzyme. That is the alkaline phosphate enzyme, which is used for the DNA washing. Now,
Okay.
the second step, that is known as the fragmentation step. In this step, what happens? We use the endonuclease enzyme, which is generally known as the chemical knives, because
Yes.
we are to go the fingerprint. So, second step into the fragmentation, the molecules, DNA molecules, broken with the help of enzyme restriction endonuclease, cuts then into the fragment. The fragments of DNA are also contains the V and TR.
Okay.
Fragments are separated according to the size by gel electrophoresis. So the third step is the electrophoresis.
Okay.
What is the meaning of electrophoresis? Electrophoresis means the separation of fragments under the electrolytic condition,
Okay.
right? So it is common. What is the charge on DNA? Do you know?
Mm. Positive.
No. DNA is negative in charge,
Okay.
right? So when we place the DNA fragments into the agarose gel, then what happens? Means into the agarose gel, after applying the electric current, the fragments are moved into the agarose gel,
Yeah.
right? During the motion, what happens? The short term or short length of the DNA fragment, they moves away, and
Okay.
lengthy DNA strand that is found remain near to the well, right?
Okay.
This is known as the DNA fingerprinting, electrophoresis. Then the fourth step, the separated fragment of the single-stranded DNA are transferred onto a nylon membrane. This step is carried out when what happens? When we do the electrophoresis, during electrophoresis means the fragments are moved into the nylon, fragments are moved into the agarose, and into the agarose bands formation occur of the fragments.
Base?
Band formation.
Okay.
Right? That bands are impermanent because agarose gel is a semi-solid in nature, and when any shaking occur into the agarose gel, then the bands get disturbed.
Okay.
So for avoiding this, what happens? We do the blotting process and separated fragments of the single-stranded DNA. They are transferred on the nylon membrane.
Okay.
That is known as the blotting. Right?
Yes.
After blotting, what happens? On the nylon cloth, the DNA band, they become permanent. They never moves. And each strand of DNA, single strand of DNA, they form the permanent band on the cloth.
Yes.
Confirm? Now next step, that is the probing. What is the probing? Radioactive DNA probes have been repeated base sequences complementary to possible VNTR as poured over the nylon membrane. Probing is also most important.
Yeah.
Right? Why we do the probing? Because we get the sample from the crime place, and that samples, okay, they are obtained from the criminals. So we need to know the real DNA strand which obtained from the crime place.
Yes.
तो हम यहाँ पे क्या करेंगे? प्रोबिंग करेंगे, मार्किंग करेंगे कि ये जो डीएनए हमको मिला है वो क्रिमिनल का डीएनए है. राइट? इसके लिए हम क्या करते हैं? वी यूज द रेडियो एक्टिव मटेरियल्स. दैट रेडियो एक्टिव मटेरियल्स इट गेट इल्यूमिनेटेड व्हेन वी pour the एक्सरेज.
ओके.
पता चल जाता है कि ये सैंपल मिला था और ये हमने टेस्ट किया सस्पेक्टेड पर्सन का डीएनए, और ये दोनों ये हमको क्रिमिनल का है और ये सस्पेक्टेड पर्सन का है. अगर दोनों मैच करते हैं तो सस्पेक्टेड पर्सन इज द क्रिमिनल.
Yes.
If are not matched, then what happens? Suspected person is not criminal. So here the radioactive probing, that is also important. Some of them will bind to the single-stranded VNTR. The method of hybridization of DNA with probe is called the southern blotting. तो हम साउथर्न ब्लॉटिंग करते हैं. कैसे करते हैं प्रोबिंग के लिए, उसको हम आपको दिखाते हैं कि साउथर्न ब्लॉटिंग कैसे करते हैं. राइट है? ये साउथर्न ब्लॉटिंग का पूरा सेटअप है. ओके. यहाँ पे ये जेल रखा हुआ है. राइट? ये नायलॉन मेम्ब्रेन. ओके. ये नायलॉन मेम्ब्रेन है. राइट? और उसके ऊपर ये पेपर टॉवल रख करके हम क्या करते हैं? उसको ब्लॉट का सेटअप करते हैं. ओके. जब हम यहाँ पे सेटअप ये देते हैं तो उसमें कैसा होता है कि जो प्रोबिंग करने के बाद जो हमारा रेडियो एक्टिव मटेरियल होता है उस पर क्या करता है? वो जाकर के सेट हो जाता है. एंड वी फाइंड आउट द मीन्स वी कैन डिस्टिंग्विश बिटवीन द criminal DNA and the suspected person DNA by the probe. Yes. Right? Yes. So probing is the important. Next one, the nylon membrane is washed to remove the extra probes. कि अब हमने probing कर लिया, blotting किया and probing किया. Blotting, probing and the electrophoresis, these three are the important steps, biological step into the DNA fingerprint. Yes. After then what happens? When we probed it and blot it, then the extra DNA probes which is found into the too much quantity, which is not sticks on the nylon cloth, that can be washed to remove the extra probes. Okay. The last step, to take an X-ray film to expose the nylon membrane to mark the place where the radioactive DNA probes have been bound to the DNA fragment. And these places are marked as a dark band. When the X-ray film is developed, this is known as the autoradiography. तो हम autoradiography करके उसका क्या कर सकते हैं? फिंगरप्रिंट देख सकते हैं. राइट है? Yeah. So after fingerprinting, what happens? When the both means the band of suspected person and band of the criminal DNA both are matched with each other. They come into the contact by the two way, means ये और ये दोनों match कर गए. तो हम उसको क्या बोलेंगे? The suspected person, that is the means suspected person, that is the person who do the crime. Confirm?
Yes.
Now see, here the whole techniques is given one by one. First is the blood sample. After the second, we find out the extra DNA, then cut into this one, then do the gel electrophoresis, then here
Yeah.
transfer on the nylon papers, then the probing occur, and after then the excess probe is removed, then X-ray and X-ray matching here. If you see the 11th number, 11th number of the step,
Yeah.
then here the matching is found like this one. आपको दिख रहा है 11th वाला?
Yeah. Yeah.
तो यहाँ पे देखिए कि ये मैचिंग को दिखा रहा है कि अगर मैचिंग में बैंड ऐसा दोनों साथ में दिखाई देते हैं, राइट है ये चीज को,
Yeah.
तो सस्पेक्टेड पर्सन जिसका डीएनए सैंपल हमने कलेक्ट किया है और क्रिमिनल के डीएनए से वो मैच कर रहा है.
Okay.
अगर ऐसा नहीं मिलता है, फ्रेगमेंट्स आर फाउंड इनटू द डिफरेंट पैटर्न, अलग अलग दिखती है, राइट है, तो उनको हम क्या बोलेंगे कि सस्पेक्टेड पर्सन इज नॉट द क्रिमिनल, क्रिमिनल इज द अदर. सो दीज आर द होल स्टेप्स. एनी डाउट इनटू द अंडरस्टैंडिंग द स्टेप्स?
नो.
वंस टाइम यू सी चेक अप द ऑल स्टेप, आप देख लीजिए एक बार. एनी क्वेश्चन? Do you want to ask any question related to this?
No.
No. Okay.
No questions.
Now come on the other points. Application. At the last, we do the application. DNA fingerprinting application. First one is the forensic investigation. As you know, into the any crime place, the postmortem report, that is the solid report. Okay. During the several times, what happens into the postmortem report? The doctors take the some bribes and they give the faulty report.
Okay.
But in the forensic investigation, when the police department or the court take the decision to find out the DNA record, he sends the DNA record into the various labs and match to the DNA record of the reputed labs.
Okay.
And he sent to the different sample to the different labs.
Okay.
Right? So, do you listen to any lab which is carrying out the DNA fingerprinting test into the Mumbai or into the India? Do you know do you listen the name of lab?
I haven't heard any names, no.
You don't know any names. See here, just I am telling you, reputed lab of the India, which is the most important reputed lab, that is the CCMB Hyderabad. Okay.
CCMD.
Yeah, Center of Cell and Molecular Biology, Hyderabad. It was established by the government under the guidance of Professor Lalji Singh, who was known as the father of Indian DNA fingerprinting.
Okay.
Professor Lalji Singh, he was the very famous scientist of the DNA fingerprinting in India. He founded to the DNA fingerprinting into the India. And he was the director of the CCMB Hyderabad. He find out the technique to do the DNA fingerprinting by a poisonous snake that is known as the crate.
Yeah.
Female crate से उन्होंने ये DNA fingerprinting की criteria को start किया. And it is also important question. Several times it is asked into the examination. So उन्होंने start किया. So forensic investigation, that is a very important technique to find out the criminals. Okay. Okay. And before what happens in the judiciary cases, several times the trial goes continuous. It takes the more times, and the criminal is the another person, a suspected person. They come into the trials, and court suffers from the difficulties to find out the result. Okay. So after the full invention of the DNA fingerprinting, court take the decision to first of all do the DNA fingerprinting, then come into the court. After 2 to 3 trials, 2-3 ट्रायल चलने के बाद अगर क्रिमिनल कंफर्म नहीं हो रहा है तो उसके लिए अपन क्या करते हैं कि फटाफट वो कर लेते हैं कि नहीं चलो आप डीएनए फिंगरप्रिंटिंग करके आओ, राइट है, तो बिकम कंफर्म. Second one, that is the paternity testing. Paternity testing means parental testing. Suppose any crime case, any case comes into the judiciary in the court, okay, for finding out the real mother or father. So before this, what happens? The court take the decision on the basis of blood testing.
Okay.
Blood grouping was the most important method, right? To find out the parent, father and mother, right?
Okay.
Mother is always confirmed. Okay. Disputes are about the father. So blood grouping was the means biological support for finding out the parent. But after the finding out the DNA fingerprinting, it become very helpful into the cases of the paternity testing. Right.
Okay.
Third one important, identification of any persons and records. किसी का record रखना है, like into the army, like into the police record, okay, like into the medical department. Several times what happens, the cases occur into the crime during the wars, during the any police crimes, right? So these departments, they started to keep out the record of their soldiers, record of their staffs on the basis of DNA fingerprinting. As you know, presently the Aadhaar, you know, Aadhaar card, that is used for the biological record.
Yes.
Right?
Yes.
But into the US, army, government take the decision to keep the all record of their army person on the basis of their DNA.
Okay.
Why?
So that no one can come in or go out in the army.
Yeah, several times what happens during the war time, some soldiers, they escape, they thrown their weapons and they disappear from the war, right?
Okay.
And during the war persons, generally the means you can say the soldiers, they collect to the some samples of the, you can say the bunch of the muscles, bunch of the body parts to give their parents. Okay, that is the, it is a body part of your member, right?
Okay.
During that times कि कभी कभी ऐसा होता था कि वार में जो है, जो सोल्जर्स जाते थे लड़ने के लिए, वो अपने वेपन को छोड़ दिए और वहां से भाग गए. तो अब वहां पे जो मांस के टुकड़े मिले, डू यू अंडरस्टैंड द हिंदी?
या.
तो वहां पे जो मांस के टुकड़े मिले, जो ऑर्गन मिले, तो उससे कैसा होता था कि उनको लाके दे दिया जाता था, भैया आपका आदमी है और ये खत्म हो गया अभी. बट वो पर्सन, वो सोल्जर तो वहां से निकल जाता था, भाग जाता था. तो उसको रिकॉर्ड के लिए यूएस आर्मी ने क्या किया कि आपके डीएनए रिकॉर्ड में लाए जाएंगे, जो वहां से मांस के टुकड़े मिले हैं उससे उनके रिकॉर्ड को मैच किया जाएगा, राइट? And if the candidate is really died during the war, then you can means take the all available facilities which was given by the government.
Okay.
Right? So यहाँ पे इस चीज को किया गया कि पर्सनल रिकॉर्ड के लिए, पर्सनल रिकॉर्ड के लिए जो है इसको रखा गया. आइडेंटिफिकेशन ऑफ एनी पर्सनल रिकॉर्ड, पुलिस डिपार्टमेंट, मिलिट्री डिपार्टमेंट, आर्मी डिपार्टमेंट, नेवी, एवरीवेयर where the most important persons they are working, like into the banks, who are the means who are on the top level, directors, okay, secretaries, जो बड़े बड़े लेवल पे हैं, उनके सबके डीएनए रिकॉर्ड गवर्नमेंट रखती है, राइट है? तो ये दिस इज आल्सो बिकम इम्पोर्टेंट टूल्स. नेक्स्ट फोर्थ वन, दैट इज द फाइलोजेनेटिक रिलेशंस. Phylogenetic relation, what do you know the meaning of phylogenetic?
No.
Phylogenetic means the arrivals from the old into new. It is a relation of individual, because we are at the present time and we are evolved animals from our ancestors.
Yes.
If we see the ancestry, then what happens? All the animals which are present on the earth, they are evolved from the old animals timely. Periodic
Yes.
evolutions occur. And the
Yes.
character which is found into the unicellular, multicellular organism, all the characters, they are present into our body. So
Yes.
this relation is known as the phylogenetic relations.
Okay.
And phylogenetic relations that can be also studied on the basis of DNA profiling,
Okay.
because the DNA of the unicellular animal, multicellular animal have not any change. All the nitrogenous bases, adenine, guanine, cytosine, thymine, right? They all
Yeah.
are present into the same manner in the unicellular and multicellular.
Yes.
Only difference occur. So can you tell me the difference? What is that difference which I am indicating to you?
No, I can't recollect.
The difference occur into the length of DNA.
Okay.
Basic components of DNA that are same, means sugar is present, nitrogen base are present, phosphate molecule is present, but the length of DNA that become very,
Different.
yeah, it is different. In unicellular, the length of DNA is small. In multicellular, length of DNA is large, and it varies according to the phylum. From means protozoa, porifera, coelenterata, platyhelminthes, Helminthes, till the invertebrates means chordata and mammalia. The length of DNA is varies, but their
Okay.
basic components that are same.
Yes.
Next one, last one, that is the research and evolution. That has also become the subject. How does this research and evolution goes? Because the evolution is the process, slow process to development of organism,
Yes.
and research are going several times. The scientists take the decision to find out the what is the ancestry of human, what is the ancestry of the mammals, what is the ancestry of the birds. So all
Yes.
the answers question of all the answer of these questions, they comes through the DNA fingerprinting. If we know the best criteria of DNA fingerprinting, DNA profiling, we can know about the everything,
Okay.
right? So these are the
Yes.
topics. Here are the some samples, means diagrams which generally indicate about the means criminal investigation. ये कैसे दिखता है, यहाँ पे ब्लड स्टेन लेते हैं, इसको आपको ड्रॉस करते हैं, देखते हैं कि मैचिंग अगर इस तरीके से दिखाई देती है, यहाँ पे देखिए मैचिंग दिख रहा है आपको, हाँ, ये मैचिंग दिख
Yeah.
रहा है, सॉरी, ये मैचिंग दिख रहा है, यहाँ पे ये मैचिंग दिख रहा है. तो these matchings they indicate about the means criminals.
Okay.
Here the paternity test is given, means sample collected from mother and father, isolate the DNA, and here the matching that can has been given. After then,
Okay.
what are the research into the botany? Botany में क्या research हुए हैं? With the help of DNA fingerprinting, an essential tool for genotype of identification in both wild plant and cultivated species.
Okay.
DNA profiling is used for protection of biodiversity, identifying marker of the traits,
Okay.
identification of gene diversity and variation.
Okay.
Identification in plant always been an issue for the botanist because of the large variability of the composition and relative amount of chemicals in particular species of plant